Hello everyone, welcome back. We all know the current pandemic situation, right? Apart from this pandemic, we could see the emergence of many different microorganisms. There are cases reported with mucormycosis, which is a fungal disease, and now there are cases that are reported with this Zika virus, especially in the state of Kerala. So today in this video, let us discuss about this Zika virus in detail. This Zika virus is a mosquito-borne disease. These mosquitoes are the dangerous creatures which are responsible for causing many dreadful diseases. For example, if we take malaria, that is caused by a mosquito and there is no vaccine available against malaria till now. And the other diseases like dengue, chikungunya and also West Nile disease, all these diseases are caused by a mosquito. Even this Zika virus is also caused by a mosquito which belong to the group of flavivirus. And those other mosquito-borne diseases of dengue, chikungunya, West Nile, yellow fever, all these viruses belong to the group of flavivirus. These flaviviruses are the tiny structures that generally consists of RNA, protein and a lipid membrane. Right, here the RNA is a genetic material. Certain viruses carry DNA as a genetic material, whereas some other viruses will carry RNA as a genetic material. Here this Zika virus carries positive sense single-stranded RNA as a genetic material. Generally, if we want to define a virus, we simply say that these viruses are the nucleoproteins. That means they contain a nucleic acid, whether it could be a DNA or a RNA, that is surrounded by a protein coat called capsid. And there are certain viruses which contains an additional covering called envelope, whereas certain virus doesn't contain any envelope. We call those viruses as non-enveloped viruses. When we talk about this Zika virus contains positive sense single-stranded RNA as a genetic material that is surrounded by a protein coat called capsid, and it contains an additional layer called envelope. So this is the capsid and this is the envelope. This is the genetic material, that is RNA. Some viruses will carry negative sense RNA and some viruses will carry positive sense RNA. Positive in the sense they will act directly as mRNA and they can undergo the process of translation giving rise to a protein. But if a virus carries a negative sense RNA, first it should convert into positive sense, and that positive sense RNA will act as an mRNA, followed by a process of translation, it will give rise to a protein. And moreover, these viruses are obligate intracellular parasites. Obligate means strictly these viruses require that particular condition for their survival. Intracellular parasites, that means these viruses require a host for their replication. They won't replicate outside the host. They remains inactive. Once this virus enters into a cell, then it becomes active. It will carry out its life cycle, giving rise to more viral progeny. And this newly formed viral particles will come out of the cell and they are ready to infect the new cell. Right? So that is the reason we call them as obligate intracellular parasites. Parasites means which depend on others for their survival. Right? So here these viruses will only replicate after they enter into a cell. This Zika virus, which belonged to the group of flavivirus, was first identified in the Zika forest of Uganda in the year 1947 in a rhesus monkey. That is the reason this virus is given with the name called Zika virus. Why? Because it was first identified in the forest of Zika. Later, it was identified in humans in the year 1968 for the first time in Nigeria. As I already mentioned that this is a mosquito-borne disease. This disease is transmitted from one person to another person through bite of an infected Aedes mosquito. And when a person is infected with this virus, he usually exhibits the symptoms of mild fever, rash on his skin, conjunctivitis and also muscle pain. As these viruses are not capable of replicating outside the host, This Zika virus replicates within the host of several species including humans, mosquitoes, and also in monkeys. Coming to the structure of Zika virus, it is an enveloped virus and this envelope contains E and M proteins. Moreover, this Zika virus exhibits icosahedral symmetry and it contains positive sense single-stranded RNA as a genetic material. The virion is approximately 40 nanometers in diameter with surface projections that roughly measure around 5 to 10 nanometers. And the nucleocapsid is around 25 to 30 nanometers in diameter, surrounded by a host membrane derived lipid bilayer. Coming to the epidemiology, as I already mentioned that this virus is first identified in the Zika forest of Uganda in rhesus monkey, that is in the year 1947. Later in the year 1968, it was first identified in humans in Nigeria. First major outbreak with 185 confirmed cases was reported in the year 2007 in the Yap Islands. Later in the year 2015, first cases confirmed in Brazil, and this country has experienced the largest epidemic ever, recorded with 4,40,000 to 13 lakh suspected cases that are reported by the Brazilian health authorities. There are around 3,174 cases and 38 deaths from microcephaly in Brazil as of 21st October 2015. There has been total deaths of 152 as of 21st October 2015 in the world. Right now in this year 2021, there are cases reported in the state of Kerala. Coming to the signs and the symptoms, symptoms of Zika virus are mild and they usually last for 2 to 7 days. Symptoms include headache, mild fever, rashes on skin, pink eye, conjunctivitis, muscle and joint pain, and also malaise. These symptoms are quite similar with dengue or chikungunya. If you observe a patient suffering with dengue virus, he will exhibit the symptoms of severe joint pains. We can also call dengue as a breakbone fever. Person who is suffering with those diseases will exhibit severe muscle pains and severe joint pains. That is one of the important symptoms that a person exhibit when he is infected with flaviviruses, whether it could be a dengue virus, chikungunya or a Zika virus. Coming to the transmission of Zika virus, this Zika virus is transmitted by an infected mosquito called Aedes aegypti. And this virus is also transmitted by another mosquito species called Aedes albopictus, which we generally call it as Asian tiger, right? So these two are the mosquitoes which are mainly responsible for transmitting this Zika virus. These infected mosquitoes will feed on a blood meal of a human being. As these mosquitoes carry this Zika viral particle in their saliva, they are going to deposit this virus into a human being. This is how they usually transmit the virus into a healthy human being. Now this virus will gain entry into a human being. It will carry out its life cycle, giving rise to more viral progeny. So what happens at the end of the life cycle? There will be a release of more viral progeny. We call them as new virions, which will come out of the cell and they are ready to infect the new cells. It was also reported that this virus is transmitted through blood transfusion and also through sexual contact. Moreover, in the year 2015, it was reported that this virus is present in the amniotic fluid of a fetus. That indicates that this virus can cross the placenta and it is going to infect the fetus from a mother. But till now there are no reports that this virus is going to be transmitted from a mother to a baby through breastfeeding. After this step of transmission, now what the virus is going to do after it gains entry into a cell? Now the virus is going to infect the individual cell of a human being and it will carry out the life cycle. So at the end of the life cycle, what happens? There will be increase in the viral progeny, right? So the population of this virus will be increased. The new virions will be released out from the cell and they are ready to infect the new cell. If you look at the picture, as I already mentioned that this Zika virus contains some proteins on the envelope, right? So those proteins are useful in binding the virus with the host cell. See, these are the receptors. So with the help of the receptors, this virus is going to bind with the human cell. And after the step of binding, now this virus will gain entry into the cell. After the virus gains entry into the cell, now it is going to release the viral RNA. When it undergo the process of replication, it is going to form more copies. And as I already told you that this virus contains a positive sense single-stranded RNA, it can directly act as mRNA and it will undergo the process of translation giving rise to a protein. See, here the RNA is present in the cell cytoplasm, right? It is going to use the machinery that is required for the synthesis of a protein. Now, when this RNA undergo the process of translation using the enzymes that are present in the cell, it is going to give rise to a long protein. This synthesized long protein is chopped into several smaller proteins, among which some of these proteins will code for the structural proteins which are necessary to form a new viral particle. In order to form a new viral particle, again it should have some structural proteins, right? And moreover the genetic material RNA, right? So here this long protein that has been synthesized by the process of translation will be chopped into a smaller proteins, and these proteins will code for the structural protein of a virus. This so formed proteins and also the copies of the genome will assemble together at the surface of endoplasmic reticulum. See, this is the endoplasmic reticulum and this is the Golgi apparatus. This assembled viral particles will bud off from the endoplasmic reticulum. During this process, some part of the membrane will be taken up by the virus. So that membrane is nothing but the envelope of a virus. Now these viral particles will travel through the another structure called Golgi apparatus, where they will undergo further processing and finally they will release out from the cell. See here, observe the picture here. They are coming out from the cell. So these are the new viral particles and now they are ready to infect the new cell. Coming to the pathogenesis, the incubation period of this virus in mosquitoes is usually 10 days. Whenever this infected mosquito bite a human being, now the virus is transmitted from a mosquito to a healthy human being, right? So what are the primary vertebrate hosts here? Monkeys and human beings are the hosts, right? So whenever this mosquito feed on a blood meal of a human being, now the virus will gain entry into a human being, right? The pathogenesis of the virus is hypothesized to start with an infection of dendritic cells near the site of inoculation, followed by the spread of virus to lymph nodes and into the bloodstream. So once the virus enters into the human body, initially it will infect the dendritic cells. From there it will reach the lymph nodes and it will enter into a bloodstream. Generally, all the flaviviruses will multiply in the cytoplasm. But it was observed that this Zika virus is also present in the infected cell nuclei. Infection with the virus appears to be linked to the development of usually small heads and brain damage in the newborns, which we call it as microcephaly. Because this virus can cross the placenta and it can infect the fetus. It is going to damage the brain. That we call it as microcephaly. So this disease is especially dangerous in a woman who are in the first trimester of a pregnancy. Coming to the diagnosis of Zika virus, PCR, which stands for polymerase chain reaction, and serological tests are especially used in the identification of this Zika virus. PCR can be used to identify the viral RNA, which is the genetic material of this Zika virus. And this PCR method is also used to identify specific viral antigens. But this PCR test is to be done only after the onset of symptoms. Whereas coming to the serological test, these tests are based on the detection of specific antibodies. And this test is to be done only after the 5 days of infection. Because once this foreign particle enters into our body, what is the foreign particle here? Zika virus. When it enters into our body will recognize this virus as a foreign particle and it will start producing the antibodies, right? So our body will take some time to produce antibodies. If we wait for some 5 days and if we carry out this test, we can easily detect the presence of antibodies that are specific towards the Zika virus. Coming to the treatment, there is no specific treatment or a vaccine that is available against Zika virus. But if there is an availability of vaccine, we can provide a long-term immunity to an individual. Interestingly, there is a vaccine that was developed against dengue virus, which belonged to the group of flavivirus, and that vaccine was approved for its use in Brazil. And that vaccine is prepared with the inactive version of yellow fever virus. Again, this virus belongs to the group of flavivirus. If we can apply this strategy, we may successfully develop a vaccine against Zika virus. Coming to the other important segment, that is prevention. As we always believe that prevention is better than cure. So the very important preventive measure is to avoid mosquitoes. Protect yourself from mosquito bites. You can use mosquito repellents or you can spray some insecticides to kill the population of insects, and you can use some mosquito nets. So only thing you need to do is you need to avoid the mosquito population, and moreover you need to remove the stagnant water, which is an excellent source for the mosquito breeding. Just remove the stagnant water, protect yourself from the mosquito bites so that you can avoid this Zika virus. This is all about the Zika virus. We are already into the rainy season. Please protect yourself from the dreadful diseases that are caused by the mosquitoes. Soon I'll come up with my next video. Thank you so much.